The gut microbiome can be defined as the diverse community of microorganisms, bacteria, viruses and fungi, residing mainly in the gastrointestinal tract, more specifically the large intestine. These microbes are metabolically active. They ferment fibre to short-chain fatty acids, synthesize vitamins, modulate immunity, influence the gut barrier, and communicate bidirectionally with the brain, known as the gut-brain axis. For athletes, these functions affect energy availability, recovery, illness risk, and even mood and perceived exertion.
Hello and thanks for joining. This higher level video examines how the gut microbiome affects digestion, metabolism, immunity, the gut-brain axes and athletic performance. Throughout the video, we will define the microbiome, explain key metabolites and mechanisms such as short-chain fatty acids, and describe factors shaping the microbiome. Additionally, we'll then present evidence linking microbes to performance and recovery and outline some safe, practical interventions, as well as reviewing the assessment specifics for this content in Topic A. The higher level content covered in this video should support with MCQ and extended response style exam questions, which you may come across. If you manage to stick around until the end, I'll reveal some IB exam technique and linking questions related to the gut microbiome. Don't forget to keep spreading the word with colleagues and peers, to like and subscribe to the channel. Slowly but surely, we are converting short-term viewers into long-term subscribers. Thank you in advance for your support. And of course, I'll continue producing and uploading free sports science content each and every week for you. With that being said, use the timestamps to jump to specific sections which interest you. And now let's get stuck into the video.
The objectives you should achieve by the end of this video include being able to explain the role of the gut microbiome. Certainly not too many objectives to cover for this HL content, but this will allow us to spend a little more time looking at the influence of the gut microbiome on health and performance. Gut microbes break down dietary fibre to make short-chain fatty acids. These feed colon cells and helps keep the gut barrier strong. Some enter the bloodstream and affect liver metabolism and how tissues use fuel. The short-chain fatty acids reduce inflammation and improve insulin sensitivity, which supports general metabolic health and can help endurance performance by improving substrate use. Some gut bacteria can use lactate and convert it into other metabolites. For example, Veillonella species turn lactate into proprioset. Early research suggests this pathway might help endurance by providing extra fuels and reducing local lactate buildup. The microbiota also makes certain B vitamins and vitamin K and influence how minerals like iron and calcium are absorbed. These functions matter for oxygen transport and bone health, both important for athletes. A healthy microbiome trains the immune system and preserves the gut barrier. When the microbiome is out of balance, known as dysbiosis, gut permeability can rise and let bacterial products enter the circulation, which raises systemic inflammation and can slow recovery and impair performance. Finally, the gut communicates with the brain. Microbial metabolites and signals traveling via the vagus nerve influence neurotransmitters and the stress response known as the HPA axis. This gut-brain link affects mood, perceived effort, stress resilience, and cognitive recovery after hard training. Diet is the biggest influence on the gut microbiome. Diets high in diverse plant fibre increase microbial diversity and production of beneficial short-chain fatty acids. Fermented foods like yogurt or kimchi supply live microbes, and prebiotic fibres feed beneficial bacteria. By contrast, highly processed, high-sugar diets reduce diversity and encourage more pro-inflammatory communities. A practical tip: athletes should prioritize a variety of fibre sources for gut health, while still timing and periodizing carbohydrate intake to meet training needs. Exercise itself changes the microbiome in helpful ways. Regular physical activity, both acute sessions and chronic training, tends to increase diversity and support metabolic and immune health. Medications can strongly affect the microbiome. Antibiotics can disrupt gut communities deeply and sometimes for a long time. Other drugs like NSAIDs can also alter composition and gut barrier function. After antibiotics, focus should be on dietary recovery, including fibre and fermented foods where appropriate. Sleep and stress matter too. Poor sleep and chronic stress lower microbial diversity, and can weaken the gut barrier through stress hormones like cortisol. Good sleep and stress management strategies support the microbiome and recovery. Early life factors such as mode of birth, infant feeding, and genetics help shape the initial microbiome and host microbe interactions, but ongoing environment and diet remain highly influential across life. Research shows links between the gut microbiome and athletic performance, but most findings are early and mostly associative. Observational studies find that many elite endurance athletes have greater microbial diversity and more bacteria that make short-chain fatty acids or metabolize lactate. For example, higher levels of Veillonella have been associated with better treadmill performance, and animal studies suggest converting lactate to propione might help endurance. These results are promising, but do not prove cause and effect in people. Some studies also link certain gut communities with better fat oxidation. That could help endurance athletes by sparing glycogen and prolonging performance, but the evidence in humans is not yet strong. A healthier microbiome is often associated with fewer illnesses and quicker recovery, probably because of better mucosal immunity and lower inflammation. Still, most human data are correlational. Specific probiotic strains can have benefits, but effects are strain and context dependent and need confirmation in athlete groups before routine use. For interventions, start with diet, eating more fiber from whole grains, legumes, fruits, and vegetables to feed microbes that make beneficial short-chain fatty acids would be worthwhile. Include fermented foods like yogurt, sauerkraut, kimchi to add live microbes and increase diversity. Add polyphenol-rich foods such as berries, tea, cocoa, which also support helpful bacteria. Keep carbohydrate timing matched to training. Fuel well for high-intensity sessions, but maintain overall diet quality to support the microbiome. Supportive lifestyle habits matter. Regular, moderate exercise generally increases microbial diversity. Avoid chronic excessive training without adequate recovery. Manage stress with sleep, mindfulness or breathing techniques, and aim for 7 to 9 hours of sleep to protect the gut-brain axis and recovery. In this section, we'll take a look at 2 hypothetical case studies. Based on the content covered so far, can you identify some strategies to ensure a healthy gut? Pause the video after each case study is revealed and propose a plan. Case study 1. An endurance athlete with frequent colds. What would you suggest? A possible strategy for case study 1 to promote a healthy gut might be to start with food first, add more prebiotic fiber, whole grains, legumes, fruits, vegetables, and regular fermented foods, yogurt, kefir, sauerkraut, to support beneficial microbes, improve sleep, and reduce stress with routines or brief mindfulness practices. If infections continue, trial an evidence-backed probiotic strain under guidance and track changes in upper respiratory infection frequency alongside training load and recovery. Case study 2. An athlete taking antibiotics before an important event. What would you suggest? A possible strategy for case study 2 to promote a healthy gut might be to focus on gentle, high-quality, fiber-rich meals as tolerated and include fermented foods to help microbial recovery. After finishing antibiotics, consider a clinician-recommended probiotic. Choose third-party tested products if appropriate. Reduce non-essential high-intensity training until recovery is clear, and monitor symptoms and energy so competition preparation remains safe. Long. Okay, well done. Let's move on to some formative assessment and exam technique. In this section, we'll have a go at some quick-fire multi-choice questions based on the gut microbiome content, which may appear in paper 1A. Pause the video here, have a go and see how you get on. Let's take a look at the mark scheme so you can self-assess. Okay, good stuff. Next, let's crank it up and take a look at some data-based, short answer and extended response questions, which may appear on paper 1B and paper 2 based on the gut microbiome content. For possible short answer and extended response style IB exam answers, you may be required to explain the microbiome, explain mechanisms such as short-chain fatty acids and lactate conversion. cite evidence examples and limitations, or propose some practical interventions. On screen, you can see some short answer questions related to the video content. Use the command term key to carefully consider the requirements of each DBQ and ERQ question and construct your answers with the appropriate level of detail. Pause the video now and give them a go. Okay, let's take a look at the answers and see how you performed. Take a moment to pause the video here to reflect and self-assess. That is the end of the additional higher level content for this element. We can finish by thinking about the following linking question from the course guide between what we covered in this HL video and topic C. C, 4.2, how does anxiety affect the gut microbiome? To summarize, the gut microbiome is a dynamic, influential system that affects nutrient metabolism, immune function, recovery, and the gut-brain axis. Practical dietary and lifestyle actions can support a healthy microbiome. Targeted probiotic use may be beneficial in specific, evidence-backed cases under appropriate qualified guidance.
If this higher level gut microbiome session genuinely helped you in some way, or you would like to see more like this, please share, like and subscribe to the channel if you haven't already done so. The goal continues to focus on finding more teachers or students like you and supporting them with their IB sports science needs. Also, don't forget to click that notification bell so you're the first to know when new content hits the channel. Stay tuned for the next video where we'll look at some standard level content around energy production through the phosphagen, glycolytic and oxidative systems. Alternatively, if you would like a little refresher on previous content from topic A, I'd recommend looking at this episode right here. Thanks once again for watching, and I'll see you over on the next one.
If you made it to the end of this video and would like to support the growth of the channel, please take a look on screen now and scan the QR code. Thanks again.